If everyone has a deep breath now after that. So we have Allyson Berent, Dr. Berent here as our keynote speaker, and I could not think of a better speaker to guide us today. So Allyson is first and foremost the mother of a child with Angelman syndrome, which is quite similar to FOXG1 syndrome. She is this chief scientific director of FAST, which is the foundation for Angelman syndrome. She's really an inspiration to us. She co-founded a company called Genetics, which focused on an antisense therapy for Angelman, which was acquired by Ultragenyx and is now in clinical trials. So you have Angelman children actually in this trial. The news that we have heard about it has been pretty life-changing for the children in the trial. And so her foundation is an inspiration for our foundation, the way they think about their patient community, the way they think about their science. But Allyson in particular is definitely a mentor to us. She is truly an amazing human being. No matter what time I text her, she will get on a call, even though she has 3 children, runs a foundation, a company, etc. She's always there. And we're so grateful that she can talk to us today, and I hope she really fills you all with inspiration. She's our North Star. She's our North Star, absolutely. Yes. And she actually was here for the science day. She was here the whole day, but then had to, she's a rare disease mom, had to go back to her children in New York. So she's going to be joining us via Zoom.
Great. Allyson, do you hear us? Do you hear us?
I hear you. Can you hear me?
Okay, great. Thank you so much for being here. And let's get started.
Okay, I'm just sharing the screen. Can you all see that? Yeah, you can see it? Everybody can see it? Okay,
yeah,
great.
We could see it. Thank you. Yeah.
Wonderful. Good morning, everyone. Thanks for getting up so early. I can't see anyone in the room, so there may be one person there. If it's just Nicole and Nasha, hi. If there's many people, then I'm so honored and humbled to get to meet you. My name is Allyson Berent, and I am really happy and humbled to be able to introduce you to my daughter and my journey through drug development, which has been a relatively short one, though it feels like a lifetime, as I'm sure all of you can appreciate. So just to introduce myself, I am the Chief Science Officer for the Foundation for Angelman Syndrome Therapeutics. So this is the largest non-governmental research funding foundation in the world supporting drug development and the research pipeline to bring transformative treatments to individuals around the world living with Angelman. I also am the co-director of the Angelman Syndrome Biomarker and Outcome Measure Consortium, or the ABOM. I'm the co-director of InSync, which is the International Angelman Syndrome Research Council, where we bring together experts around the world in different areas, from neuroscience all the way through to drug development, regulatory sciences, biomarkers, outcome measures, in order to do a landscape assessment of Angelman syndrome that are not involved in the Angelman space, to be able to help guide us onto all of the gaps that we have, the things that we're missing, and the places we need to do more work. I'm a veterinarian by training and by day. I'm an internal medicine and interventional radiologist specialist. So I run clinical trials in animals with naturally occurring diseases for the last 20 years, developing medical devices for them with the ultimate goal for translation to the pediatric population, where there's a huge clinical unmet need for medical devices. So that's where I've been for the last 15 or so years. Until 2015, when this beautiful little girl on the left, Quincy, was diagnosed with Angelman syndrome. And at 6 months of age, my life changed, and I decided that it had to be easier to fix a single gene disorder than to fix a cat ureter. And so I jumped into how I can help my daughter. And so, in full disclosure, I'm the co-founder and former chief operating officer of Genetics Biotherapeutics, a for-profit company that was started out of our non-for-profit, out of FAST. And that company was recently acquired by Ultragenyx Pharmaceuticals, and it's advancing an antisense oligonucleotide for Angelman syndrome, and they're in a phase 1/2 clinical trial currently. I still serve as a consultant for Ultragenyx, so I'm still very involved with the program. I am the chief development officer for Mazi Therapeutics, a company that was really started out of the vision of what parent foundations are capable of and how to put it in the hands of of real drug developers in order to advance programs from that proof of concept through into clinical trials. And so I'm able to serve other neurodevelopmental disorders and monogenetic disorders outside of Angelman syndrome as well. I'm the consultant and I advise many drug development initiatives for numerous rare disease organizations, particularly in the neurodevelopmental space, and I am the member of the N1 Collaborative. So I'm going to start with my journey as a parent and really what it's like living with a patient. And this isn't going to be new to you guys. This is going to touch your heart, I'm sure, in a very deep way because you live this every day. So I'm not telling you something you don't already know, but I'm here to really just share with you a joint journey that we all share together. It may be a different gene, but it's not a different journey. And then I'm going to walk through the community journey from FAST and serving as the CSO for FAST, and then leading that into the industry journey of what it was like in our disease and how that transition from going from nonprofit to for-profit to acquisition, and then really financially being able to seed and accelerate drug development because of that journey. And of course, all of the pieces come together to be holistic and how that can help our children. So I'm going to start with a video, and this video is really, it was actually made by a CRO, a contract research organization that we worked with in order to advance multiple programs for Angelman syndrome, about a parent and what a parent's trying to do. And it's the journey of my family and Quincy. And I'm only sharing this because, as you all know, it's really easy for me to stand here or sit here, I guess, and talk to you all about what it's like to live with a rare disease and what it's like to take those steps. But it's far better, I think, if you see it and you see her. And I think you'll relate very much to her because I think the symptoms and the phenotype of Angelman syndrome are quite similar to FOXG1. So this is just a real quick journey for us and what it looks like a day in Quincy's life.
When you meet her, the first thing you will notice is her smile and her laugh. You'll also notice that she can't talk with her mouth. She talks with an iPad.
I like them. I love them.
Quincy has so many strengths and so many challenges. If I ask her to say hi, she opens her mouth and says, huh. She has Angelman syndrome. It is a deletion of one of her genes, and it's totally random. I had a normal pregnancy with Quincy, but we started noticing things about her that were a little bit different. It was weird how unfussy she was. She never cried, and she was incredibly smiley. But she wasn't happy when she was looking at people. She was just happy looking at the fan. We were worried something is seriously wrong with her. So we asked if we can see a neurologist. The first words out of his mouth were, Miss Berent, I have catastrophic news for you. What she has been diagnosed with is Angelman syndrome. I looked it up. What stuck with me the most was not walking, not talking, and having severe seizures. Under treatments, it said no treatments available. My head just started spinning. You know, she'll never go to college, she'll never go to school, she will never get married, she will never have kids. You know, all of the things you dream about for your beautiful newborn little girl, just taken away in a minute. And it was at that moment that I knew that I needed to educate myself and I needed to learn how to fix this. Because mommy and daddy fix everything. Bad news either paralyzes you or it motivates you. And for me, it completely motivated me to figure out a way to ensure that a treatment for Angelman syndrome gets outside of the mouse and into humans. The gene that Quincy is missing is actually present in all of us, including her, but it's silent. And all we would need to do is unsilence that copy of that gene, and that would be a mechanism in which we can actually cure this disorder. 2 weeks after she was diagnosed, a paper came out, and it was in adult mice with Angelman syndrome. And they gave these mice a drug called an antisense oligonucleotide in their brain, and it reversed many of the symptoms that the mice had. And I realized at that moment that if they could do it in mice, why can't they do it in humans? How can we get this to my daughter? So in order to get this from mice to humans, it is incredibly expensive. It requires a tremendous amount of work, and it requires a tremendous amount of expertise. Most of the time, the steps between mouse and human are done through a pharmaceutical company. We didn't have that luxury. We had a concept, and we had a proof of concept and a mission with timelines that were not negotiable. And we had passion. And we were able to bring on the best people in the world virtually to help our company of 5 drive a drug to a human clinical trial that could impact children living with Angelman syndrome. Now, after all of this, the trial is open for enrollment, and I may be able to enroll my daughter in this study. This drug that we have worked so hard for could be in the brain of little kids. This is a miracle that I can't believe I'm a part of. My hope is that this is transformative. I want to see her life improve. I want her to tell me the things that she can't communicate in any other way. But I'm mostly thinking about who she is and who she might be. I just want her to say hi. That's what I want.
Sorry, so I still cry when I watch that video, and I'm sure many of you feel those feels every day when you wake up and you live with your child that has FOXG1, which is probably very similar. And when I heard the word Angelman syndrome, the first thing is, how do you spell that? No geneticist generally gives you that diagnosis in the right way, and it usually is just the worst day of your whole life. And when you read about it, unfortunately, you read all of the things that your child will never do, all the nots. And then you have to bring your head around to say, well, what will she be able to do? What is the prognosis for survival? And how do I give her the best life and advocate for everything she's capable of? And how do I ensure she's accepted in this world of ability and this world of neurotypical, but is able to live the most fulfilled and independent life? And imagine going through all of that, which most of you have, in 30 seconds. Like your brain is just, how do I deal with this? And my first reaction is, as any parent would, I have to advocate for Quincy. I have to learn, I have to execute if I can change the trajectory of this little girl's life. And then I went through this, 30 seconds later, which I hope others didn't, because I'm not proud of it. I went through this period of saying, I can't do this. And to be perfectly honest, I don't really want to. And then it's the guilt of, what kind of mother am I? I have this beautiful career. I've been working my whole life to become a veterinarian, traveling the world, teaching new things, really at the top of my career. This is exactly where I wanted to be, married to my soulmate, have a beautiful neurotypical older daughter. And I have to think about, how am I going to handle this as a family? How am I going to handle this as a wife and a mother to her sister? How am I going to navigate this new world while I manage a full-time job in this life of therapy? And how am I going to be able to ensure that I don't give up everything I worked so hard for? And so I felt there had to be a solution to this problem. And really, knowledge is power, and I had to understand everything. And my husband and I always have this motto that things are either possible or impossible, and there's no in between. And until someone can tell me that something's impossible, I'm going to believe that it's possible. So we're going to try to fix this, and we're going to figure out a way. But I had to be brave, and I had to be smart. And so I learned everything I could about the genetics of Angelman syndrome. I learned that it was monogenetic, that it was non-degenerative, 2 very, very important factors, that it was random, that I was not a carrier for this, that if I had more children, that they would not be at risk, that my older daughter would not be at risk of carrying this to her children. And it was a really focused neurologic disorder. It only impacts neurons. And when I understood the genetics, that it affects chromosome 15, that it's very unique, and that it's an imprinted gene. So what that means is that we all have a maternal and a paternal copy, a mother's and a father's copy on the 15th chromosome of many, many genes. But there's a handful of genes on the father's copy in all of us, and me and you and your children and my children, that are silenced with this long non-coding RNA called the UBE3A antisense. And that long non-coding RNA ultimately silences the father's copy of the gene in all of us. So all of us only read the mother's copy of this gene called UBE3A. And in kids with Angelman syndrome, that gene is either missing or mutated. And if there's a variant and there's a mutation of that gene, it's not read. But because they also have a silent father's copy, like we all do, they have actually no copy being read in their brain. So they're not haploinsufficient in their brain, they're actually completely null. But peripherally, they're haploinsufficient because they do have the father's copy that's being read in every cell of the body except for neurons. And what happens when you have a single gene in a single cell line that's missing? Well, it's quite severe. You have the symptoms of Angelman syndrome, which you might be well aware of and may experience in your everyday. A universal lack of speech. Kids with Angelman syndrome don't have a single word. Life-threatening and debilitating seizures, severe developmental delay, poor motor skills and incoordination, apraxia and dyspraxia, a very unique happy demeanor. So they're incredibly happy, loving children that love sociability. They don't have autism spectrum traits generally because of their sociability, their desire to be social and be around individuals and be incredibly involved in social environments. But some of them do have some more traits that are more autism-like. The inability to live an independent life completely across the board, regardless of genotype, severe feeding issues, and there's obviously a clear, significant unmet need. And one of the first papers that I read was this paper on mosaicism. And mosaicism means that some cells of the body are expressing the gene and some cells of the body are not expressing the gene. And that's because this doesn't happen at the egg stage, but it happens during the development, usually de novo, while the embryo is being grown. So it happens at an early stage of embryonic development in the baby. And basically some of the cells wind up having a problem where the UBE3 gene is not read on the mother's copy, but other cells, the UBE3 gene is read on the mother's copy. And what that leads to is some cells have it and some cells don't. And when that happens, it gives you kind of a natural example of what it would look like if you had 5% of cells expressing UBE3A, 10% of cells, 20% of cells. And so what you could see here are individuals with 1 to 5%, up to 20. And then there's no identified mosaic children with UBE3A deficiency over 40%. And that's probably because they're neurotypical. And the clinical symptoms of an individual with 40% of UBE3A compared to 20%, compared to 10, compared to 5, you can see actually here. So you can see that the less you have, the lower your clinical score is. But what's really interesting is that individuals with 1 to 5% of UBE3A have no seizures, are ambulatory, have some ataxia, and have some speech. Individuals with 20% are a totally different child than the 5%, where they are fully ambulatory with no seizures and they can speak in sentences. And individuals that are 40% or more can be confused with neurotypical because they have very little disability, if any at all. And so what does that tell me? That tells me that a 1 to 5% kid is not Quincy. That's a kid that has far more skills and far less challenges than Quincy has, because Quincy is 0% in her neurons. And so therefore, it gives me a lot of hope that we don't need a lot to go a very long way to change the lives of these kids. And the enemy of good is better. And we have to get away from the idea that we have to get perfect distribution and perfect transduction and perfect expression in order to make a difference in the lives of children living with Angelman syndrome. And so as a clinician, I called every senior author I could on every publication that I could find. I attended every scientific conference that I could, and something was incredibly clear to me. What was most clear is that it was an incredible decade to be a mouse with Angelman syndrome. But unfortunately, it was not the right decade to be a human. They've cured Angelman syndrome in 7 different ways, in 7 different lines, at 7 different universities. But unfortunately, there was no understanding of when we were going to get this to patients like Quincy. And so translational science was outside of the wheelhouse of so many of these scientists in order to know how to get out of their lab. These are neuroscientists, brilliant neuroscientists, who are doing such an incredible job in order to understand the animal model, understand how we can rescue the behaviors in these animal models and what that might mean for humans. But who was taking that leap out of the valley of death to jump over and get to humans? And unfortunately, without this traditional pharmaceutical model, that really wasn't happening. And so we realized that we could not wait for a treatment and we needed to help shepherd one, because that's what we do for a living, my husband and I. We translate concept and bring it to patients with a disease with the ultimate goal of getting it to kids. And so I must act like a clinician. And so in 2015, I joined the board of directors as the chief science officer for the Foundation for Angelman Syndrome Therapeutics. And what I realized once I joined that board was that translational science with a therapeutic focus was their goal since 2008, far before Quincy was ever diagnosed. And there was so much infrastructure created on the scientific side, and there was so much great work that was being done by a consortium of scientists that were really there to believe in this one mission, which was to bring transformative treatments to individuals living with Angelman syndrome. And they created this incredible team with these really strong and robust and quite outside the box research initiatives. And really, they were considering all of the therapeutic strategies. And the goal was to scientifically de-risk each strategy to have a better chance for execution, understanding that different approaches may benefit different populations. And really, when we tried to understand what steps it was going to take to get to humans, that's where I felt that I could play a really big role. So we created a true roadmap to a cure in order to understand what it was going to take to get to human candidate, to get through those IND enabling studies and get to a human. And we really needed an ask because we didn't have an ask of how much money that was going to take. We heard the number, someone said it'll take $5 million to cure Angelman syndrome. Someone else wrote it would take $20 million to cure Angelman syndrome. Well, I hate to tell you, but it's probably closer to $1 billion to cure Angelman syndrome. And until you really lay out those numbers and understand that, you don't know what your ask is. And if you don't know what you're asking for, then you don't know what your goals are. And everyone wants to give money when there's a tangible goal of what those milestones are that they're going after. And so we created an ask, and the ask was to get to human candidate from the proof of concept platforms. And we were funding 6 platforms. And of those 6 platforms, in 24 months, it would take $5.8 million just to get to human candidate, not to get through IND enabling studies, which is what the FDA or various different regulatory agencies require to get to humans. And so we committed to this family that donated $5.8 million after 6 weeks of a campaign that we would do this in 24 months, which is a huge ask. We brought together the scientific team, we brought the money, and we had this ask, and we had to execute upon that ask. And so our funding philosophy is to cure AS, and we do that through collaboration, understanding, readiness, and expedition. So we're here to bring everybody to the table to understand what we need to do to get it ready for clinical trials and to expedite that drug development pipeline to get there. And so really our roadmap for success was that we can replace the missing copy of the gene or the missing protein. We could activate the silent copy of the gene by inhibiting that long non-coding RNA. We can identify symptomatic treatments because this is a synaptopathy at the neuron, and we can really understand how do we address the synapse. But we need to prepare for clinical trials by characterizing models, by creating biomarkers and endpoints, by really understanding clinical trial design and what's appropriate for this population and what's going to be agreeable by the regulators, and to really ensure that we're addressing all the needs of every genotype living with Angelman syndrome so that we do not leave a single patient behind. So in 2016, we launched the first pre-competitive consortium called the ABOM, the Angelman Syndrome Biomarker and Outcome Measure Consortium. We started with 2 pharmaceutical companies that were interested in Angelman syndrome. Within a year, it was 7 at the table, and today there are over 31 people at the table. What was most impacted in the lives of those living with Angelman syndrome is what we wanted to know. We did a family survey just on Facebook with 332 responders. And in those responses, what was very, very clear was that the number one thing that parents wanted to see improve in the lives of their children was an improvement in speech and communication, both expressive and receptive language. Number 2 was seizures, and number 3 was gait. Then we did a publication, a disease concept model, which is a really rigorous publication that took 3 years. And what that publication really supported was that speech and communication were number 1, seizures was number 2, and gait was number 3. So our little Facebook survey for free supported this multi-$100,000 disease concept model, but that's really what the FDA wants to see. So when we realized what was important in the lives of individuals living with AS, then we can work toward creating endpoints that would be measurable and valuable in a clinical trial. So we prioritize the functional domains in Angelman syndrome, like communication, gross motor, fine motor, global, through something like a clinician global impression of change. So what does the gestalt of the symptomology of Angelman syndrome look like, and how will we measure that if it changes in a clinical trial? What about quality of life and activities of daily living, looking at cognition in individuals and looking at sleep and seizure control? And so what is important to families doesn't always align with what is important to regulators and to pharmaceutical companies. And that's because what's important to us, like cognition, is not always able to be measured. And if you can't measure it, it's not a good endpoint. And so really putting together what's important to families, what's important to pharma, what is measurable, what is acceptable by the regulators, and then most importantly, what is going to be paid by the payers? What are insurance companies going to pay for? Are they going to pay for an improvement in quality of life of parents? Well, in Europe, they will. In the US, that's not clear that they actually put a lot of weight on that. And so you really have to be careful to understand your audience and understand how to get those biomarkers in order. And so we put together this dashboard for communication, fine motor, gross motor, global, activities of daily living, quality of life, cognition, sleep, seizure behavior, and then measurable biomarkers. We looked at all the ways that has been measured in the natural history study of Angelman syndrome, which had been done over a 15-year period of time, where we had all of these endpoints that were being prospectively collected on these kids. And then we looked at what was going to be acceptable by regulators and by the payers. And so you could see that really the pyramid gets more and more narrow. And what we were able to say is if we look at all of these domains, there are just a certain number of these endpoints that we need to focus on that are agreeable by all parties. And that's really where we put our money. And we invested $1 million in developing these endpoints that were pre-competitively shared with every pharmaceutical company in the space so that no one was taking this on their own and not sharing it for the better good of all of the patients. And then when we got that grant of $5.8 million at the end of 2016, 6 months later, everything changed. One of the grants that we were funding was advancing an antisense oligonucleotide for Angelman. And what we found was that we could increase the paternal expression of the UBE3A gene with an antisense oligo as we basically inhibited that antisense or that stop that was stopping the expression of that gene. So we can see something go up and something go down. And that's exactly what we wanted. And this was in the neurons of patients with Angelman syndrome. And then an animal model was done with a mouse version of this. And ultimately what they found in the mouse model, which was actually done as proof of concept before this even happened, was that we can rescue the symptoms, some of the symptoms in the adult mouse model of Angelman syndrome. And we only needed the proof of concept to say, if we knock down the antisense and we activate the gene, then we're going to see an improvement in behavior. And we had that. So we had a beautiful proof of concept. And then we had a biological proof of concept to show that if we actually increase expression of UBE3A in the paternal, you could see here that we have no UBE3A gene being expressed in the Angelman syndrome mouse model without the antisense oligo. And with the antisense oligo, you could see that we're expressing paternal UBE3A throughout the entire brain. And so what this showed us was together through some biology, through some simple proof of concept in behavior and some proof of concept in biology in humans, we had a path forward to bring a drug to our children. And so that was the impetus to start Genetics Biotherapeutics. And we started this company, a for-profit out of a nonprofit, in order to advance an antisense oligo for Angelman syndrome. And we did that through this industry journey that suddenly we were taking. So FAST launched Genetics Biotherapeutics in 2017 in collaboration with Texas A&M University, the investigator who developed the ASO, angel investors who were family and friends and business folks that were very interested in understanding how we can advance this and de-risk this further, to show interest in Angelman syndrome for pharmaceutical companies. And we had to do this through a for-profit approach because we did not have the donations that we were going to be able to support the work, which is going to be tens of millions of dollars. And so we were singularly focused to advance this program, and we brought together world-renowned consultants and experts in a virtual way, from non-clinical understanding toxicology, pharmacology, manufacturing, chemistry, regulatory, and clinical. We applied for orphan drug designation, rare pediatric designation, fast track designation. We received that through the FDA. And we did all of our work in the monkey because the monkey is genetically conserved to the human. So we can test the human candidates in the monkey model where we could not do that in the mouse model. And in 2019, after we had some very important data in the non-human primate, we collaborated this program with Ultragenyx Pharmaceutical in order to launch the phase 1/2 clinical trial, because we as a small company could not afford to do that on our own. So that was the point where we decided we needed to raise more money. And we were going to do that with either a pharmaceutical partner or with venture capital, and we decided to go with a pharmaceutical partner. After the phase 1/2 launched, Ultragenyx acquired the company in 2022, and now the company is fully in their hands, and I still serve as a consultant for that program. So that was an incredible milestone for our organization because FAST owned 58% of that company. And because of that, with that acquisition, that put money back into FAST so that we can further advance all of the other programs in the pipeline that need to get to where genetics and where the ASO got. And so really, as we think about the focus of Angelman syndrome, of the journeys of a parent, of a clinician, of a foundation, of the community, and of industry, a lot of people ask, aren't you conflicted? And I kind of think I'm the opposite of conflicted, because the best confluence of all of these agendas is what gives you the best perspective, as long as you never take the side of a single company and you always advance every program for every child. And what that means is that if your only return on investment and your most important return on investment is these children, your focus will always be right. The path to drug development requires focus, commitment, and passion, and there's nobody that has more passion than parents. This I can assure you. Nights, weekends, birthdays, and holidays, we're going to do what we need to do because we know that's going to be one less day that our kid is going to go without speaking a word. And that's all that matters. And it's hard to instill that in people that have a pipeline that you may not be the priority of, that have a lot on their plate that you may not be the focus of. And the vision of the Angelman community is what drove all of these programs to ensure that we treat all of the kids around the world living with Angelman, which is up to 500,000 kids, and we have to work for every one of them. So if we look at the Angelman pipeline 6 years ago, this is what it looked like. In academics, we had an AAV program, an ASO program, a zinc finger program, and some downstream symptomatic targets. So we were really in the basic research and drug development space. And this is where we are today. We have 4 drugs in clinical trials. We have multiple drugs in IND-enabling studies. We have multiple shots on goal in every different platform that can work for Angelman syndrome. And we have multiple shots on goal within that platform. So what we're seeing is multiple pharmaceutical companies that are working for Angelman syndrome. And these are all the programs that FAST has robustly funded and gotten them to where they are. So we did not do this just relying on pharmaceutical companies to get us there. We took the reins and we did this ourselves. And what we realized was competition breeds competition, and competition is really good for our kids. Because once people realize that it's a targetable disease, everyone jumps into the space, and that is exactly what you want. But it's not just about the drug development pipeline. We have to think about all of the things that are required to get you where you want to go, which is ultimately FDA approvals. So we have to think about animal models, registries, natural history studies, disease concept models, endpoints and biomarkers, diagnostic efforts to find as many patients as we can. The more patients diagnosed, the more appealing the disease is for drug developers. And so we really need to think about all of those efforts. And when Quince was diagnosed, that's basically where we were. We were kind of in this early stage of all of these other things. And this is where we are now. So we're really moving the mark on the roadmap in order to take this drug development pipeline to the right. And that's really where we want to be. And so when we think about all the ways that we could do this, replace the gene, replace the protein, activate the gene that's silenced, all of these companies and academics are working in order to try to help us get there. And really symptomatic treatments trying to fill any gaps that we might not get with upstream targeting. It is a holistic journey. And drug development is hard. I can assure you of that now that I've been a part of it. But I can also assure you that just like you, living with rare disease is far harder. And if anyone tries to tell you otherwise, they need to spend a single day in your life, because it is far harder to live with these kids and realize the everyday sentence that they've been given. And trying to fix it is not nearly as hard. And our foundation works really hard to accelerate and de-risk every opportunity for our children. It's very hard to rely on other parties to make your disorder a priority when the only people it's a priority to is you. And at the end of the day, money talks. Biotech is in a bad place, and they're going to drop and shelve programs. But it's not because they don't love you, and it's not because they don't believe in you or believe in your science. It's because hard times call for hard decisions. And sometimes that decision is not going to be for FOXG1 or for Angelman syndrome. And I can't say that I don't understand why they make the decisions they make. But what I can tell you is that I know why we at our foundation make the decisions we make, and that's for Angelman syndrome. So the priority for us in our community will never change. It only will change when our tagline does. And our tagline is Cure Angelman. And the only way our priority is going to change is when our tagline changes to Cured Angelman. And we're not there yet, but we're getting there. And so we are going to push as hard as we can, as far as we can. on our own if we have to, though we don't want to, and we prefer to be in collaboration with everybody that we can. And so Quincy is my reason to fight every day for a cure. And I know you have so many reasons to fight. And what I also want to tell you is that my daughter wore the FOXG1 t-shirt that I received when I was there. My daughter is sitting here screaming, wanting credit. When I received the t-shirt from Nasha, and Nicole, thank you very much, my daughter wore her t-shirt to school yesterday, and there was a QR code on her t-shirt, and ultimately her school made a donation to FOXG1, and I asked her, well, have they made a donation to Angelman syndrome? And she said, Mom, rare is one. And I said, you are absolutely right, rare is one. And I'm so glad that they made a donation to FOXG1, and we're all in this together. So anything we can do to help each other, that's exactly what we're gonna do. So remember, everything is possible until someone proves it's impossible. The boundaries are infinite. And thank you for your time. I'm really glad and hope that I could be any help to your organization.
I can turn it.